So the next talk of the section will be by Lucija Klarić, who is in the University of Edinburgh, and she will be talking about glycosylation of immunoglobulin G, as a complex trait. And Lucija, the floor is yours. You have 25 minutes.
Good afternoon, everyone. Well, good morning from UK. Believe it or not, it's as sunny as it shows here on the picture of Edinburgh. Today I'll speak about glycosylation, which is relatively new omics. We all know that in recent years, there's almost every week there's a new omics appearing in science, another omics field. But I hope by the end of this talk, I'll manage to convince you that glycomics is one to keep your eye on in the future. It's up and coming, and it's really relevant to many various aspects of biology. So to start with what glycomics is and what glycans are complex carbohydrates that are bound to the surface of proteins. Here you see several proteins, and in gray is a protein backbone, amino acid sequence. And this colorful attachments on the top, these are glycans. And as you can see, there can be many of them on many different proteins. And as you can imagine, since proteins are all about interactions, having something on the surface of a protein might influence the interaction of whatever the protein is interacting with. So glycans are shown and were shown to have a role in almost all aspects of a cell life. One very close to home current example is in the current SARS-CoV virus, which we see here. It's full of this spike glycoproteins on the surface, and these spike glycoproteins interact with ACE2 protein on the surface of our cells, and this is the way the virus enters the cell. Well, interestingly enough, both spike protein and ACE2, they both have glycans on their surface. You see this blue blobs here on the spike protein. You see this reddish blobs here on the ACE2. They are glycans. So, and not only that, currently the major risk factor for severity of the outcome is related with the locus containing ABO group. And ABO group, our blood groups are actually also determined by the glycans on the surface of our cells. So this is very fresh and new sort of a view of why and how glycans are really important in various aspects. But they have actually been around for a while, and they have been studied for quite a while. Even in 1985, there was a paper in Nature. It's a bit harder to get into Nature these days with glycomics, but we hope we'll get back there. There was a report by Parek et al. That rheumatoid arthritis and primary osteoarthritis have observed changes in glycosylation patterns in the serum of patients with these diseases. And this repeated in 1990, 1992. The area was very active in the times. They observed similar for inflammatory bowel diseases. They observed similar for systemic lupus erythematosus and many other different diseases, including cancers. And glycans were not only biomarkers of these diseases, they are also markers of the clinical courses. And it's not only diseases where the glycans are changed. We also have here example of Sean Connery, and when he was young, he had a lot of glycans with this. You see these yellow circles on the top. We call them galactoses. So we have many galactoses when we are young, but as we age, they start to reduce in numbers, and the glycan structures without galactoses increase. Interestingly, the same pattern is observed in progeria, and this is disease of early aging. So glycans are coming back to the spotlight because some now, even I think almost 8 years ago, American National Academy of Sciences issued this report saying that glycans and glycomics have a major role in almost every disease, and that we should start focusing more to truly understand the mechanisms behind these diseases. We should understand more what's happening with glycans in these diseases. So I'll be talking specifically about immunoglobulin G, and immunoglobulin G is quite important in our adaptive immune system. So when an extracellular antigen binds to the surface of a B cell, it gets sort of engulfed in the cell, and then these antigens are represented on the surface of a B cell, which then interacts with the T cell, which results in the activation and differentiation of B cells to plasma cells that are producing antibodies, IgG antibodies that are specific for this specific disease that encountered. This is, for example, a step that we all hope will works for the COVID-19 as well, that each person produces antibodies that are specific for the disease. Immunoglobulin G itself is this interesting molecule that has two regions, that has this upper region FAB that interacts with antigens, and FC region that interacts with the cells of the immune system. And interestingly, there are glycans attached, especially to this FC region, and depending on which glycans there are, the response can be either pro-inflammatory, or if you add sialic acid, the response is more anti-inflammatory, or if you remove core fucus, a specific response antibody-dependent cellular cytotoxic toxicity is started. So depending on which glycan is, what sort of glycans are predominantly bound to IgG, we would have different immune response. So how do we measure glycans? In the example of immunoglobulin G, you have a sample, person sample that contains many proteins. You first isolate IgG using protein G beads. Then you cut down the glycans from the IgG, you dye them with a fluorescent dye, and in the end, quantify them using ultra-performance liquid chromatography. What happens here that the glycans with their dye attach to the column, and as you elute the liquid of different polarity through the column, glycans of different polarity will sort of get released from the column, and in the end, you end up in the chromatogram like this. Here you see with time, each peak represents the fluorescence intensity, and each peak is actually representing a different glycan. So in the case of IgG, you end up with 24 glycan peaks, which you then quantify as your quantitative traits. So this sounds all right. What is already known about glycosylation when it's a well-known and old process? So we do know that things are already known. So we do know that as proteins pass from endoplasmic reticulum to Golgi, there's this set of different enzymes called glycosyltransferases that add monosaccharide each one at a time, or sometimes now there's more evidence that two at a time can be added. But in principle, one at a time. So we have a glycan here, and then we have an enzyme called GAT3, and it adds this bisecting glucnaq, and then you get this glycan structure bisecting glucnaq. So as proteins pass through the Golgi, we know that these enzymes create the final glycan structure. But what we don't know is how these enzymes are regulated and what are the other actors involved in glycosylation. So the aim of this project was to identify how is glycosylation IgG genetically regulated, what are these other actors, and then going step back, how is this genetic regulation related with various diseases and complex traits? And to do this, we used, to analyze this, we used the old well-known tool called genome-wide association studies. Just a brief over, or just in case, so to make sure we are all on the same page, you take thousands of people, if you have access to thousands of people, you take their genotypes. So in this case, you would have 22 chromosomes and single nucleotide polymorphisms throughout the whole of their genome. And GWAS ask a very simple question, given your genotype, so do you have A, AG, or GG? What is distribution of your trait of interest? So in this case, IGP1, we can see that people that have GG genotype have on average higher levels of the glycan IGP11. So it's a simple tool, but what it tells us, it tells us which parts of the genome are involved, associated with your trait of interest, and then we can take this forward to learn more about biology behind your trait. So we applied this tool, GWAS, on 8,000 samples from mostly UK, Croatia, with additional some sort of 3,500, 4,000 samples for replication. So we had the genotypes of these people, 2.5 million SNPs. On the other hand, we had 77 UPLC IgG glycans, so we have 24 measured here, but we also defined so-called derived traits where we just say, okay, how on average, what's the percentage of all glycans that have this red triangle, which is called core fucus. When we run this, we end up with this is a Manhattan plot of, I call it summarized, summary Manhattan plot of this study. So what it contains, this plot is superimposed 77 different GWASs. And it tells us there are 27 regions of the genome that are associated with their trait. Now the question is, all these loci, 27, they may contain many genes. So in this case, I think there were around 100 genes within the regions of 27 loci. So the next question was, how do we know which genes out of these hundreds are more likely to be involved in your phenotype? So to assess this, we use the following strategy. We first checked are the associated SNPs maybe in some genes, are they damaging protein structure and function? So are they in the coding regions of the genes? And if there's a gene that has a SNP associated as a coding, we would say that's more likely than to be associated with a trait, because it has a relatively direct explanation of how that would happen. The other level of prioritization was co-localization with expression SNPs. So are glycan-associated SNPs also SNPs that are associated with expression of genes in B and T cells? Remember, these are the cells where they're important for synthesis of IgG, or in expression of just genes in whole blood. Next level was to check if there are any genes that are co-expressed in biological pathways. Mostly they would be related with immunity. And if nothing else worked, we just reported the nearest gene. Now we have a look of our gene prioritization here. This is the same Manhattan plot, but flipped a bit differently. Here on the bottom, so this is not the first US of IgG. So here on the bottom, you see already known and already reported associations. But on the top, you see the novel associations. And there are 14 novel loci that we discovered in this study. And you see here are glycosyltransferases. The enzymes that I mentioned are part of glycosylation pathway are here. But what's also interesting to see that we now have a whole number of other genes, other actors and other players that might explain how this glycosyltransferases are regulated by these other genes. So to do this, we did the following. This is now a list. So if you wanted to do experimental study, validating any of these hits, we would suggest to go with those that have multiple levels of evidence suggesting that the gene is actually associated with your trait of interest. But now the main question is from the start was, how are these glycosyltransferases regulated? So to do this, we thought about this. So let's assume you have this MGAT3, that's one glycosyltransferase, and this is its effect of the SNP in this MGAT3 locus on in this case, it's probably like 30 different glycan traits. So this is just the effect of MGAT3 SNP on IgP44 and IgP50 and so on. Our main assumption was if there are any other loci that have almost the same pattern of association through all the glycan traits as glycosyltransferase, well, we could assume that then this locus is somehow regulating expression of glycosyltransferase itself. So if you look at these two nearby, one on chromosome 22 and one on chromosome 21 loci, you see indeed they have opposite colors. So red is positive, blue is negative. But indeed, they do have almost exactly the same patterns. So what now we could suggest that MGAT3 is regulated by these two other loci, one of which is chromatin remodeling protein, SMARCB1, and another one is RUNX1 transcription factor. Interestingly, there is a binding site for RUNX transcription factors, and SMARCB1 was shown to interact with RUNX1 in regulating transcription. So doing this is just a small example. We did this on all 27 loci. Now this is just expanding. Now you have 27 columns here and all the glycan. And what we did, just simply calculated pairwise loci effect Spearman's correlation. So just Spearman correlation between these vectors of locus effect on all glycan traits. And doing this, we managed to enable us. Sorry, I'm not sure what happened. I'll try again. Yeah, sorry about that. Apparently there's a combination of keys that just exited the screen. So this enabled us to create this network of genes. Now here circled are glycosyltransferases. And now we can go and suggest specific connections between our genes and other genes and glycosyltransferases. The thicker the line, the stronger the correlation. So here, for example, correlation is almost 1 between these two effects. So, of course, this is just a data-driven network. Can we validate it somehow? First, what we did, we looked in the string protein-protein interaction network. And you just submit your list of genes and see which genes are part of the different networks. And it came up with two networks. One is glycosylation. So you see here we have exclusive glycosyltransferases, and this is based on the CAC pathway of glycosylation. And then there was this other network which is transcription related. So there are many transcription factors that are regulating themselves, and many of them are important in B cell development. How this compares to our network? It compares in a way, for example, you see the red connections here. These are the connections that are discovered in the string database as well. One important thing to note is that in the string, if you look in the string itself, you can have this glycosylation network completely separated from the transcription network. So what we did in this case with this our new method, the data-driven method, is brought the connection between the two parts, how to connect the two parts of the different networks. Of course, this is some level that our network is probably not just random noise or by chance, but we of course try to validate some parts of this network in the lab. So we took this part, I-1 transcription factor, FAT8, and I-3 transcription factor, and we went to the lab. Well, our colleague Chloe Stanton did this part. And so she first asked the first question, does I-1 bind to FAT8 regulatory region? And she used chromatin immunoprecipitation and really showed that I-1 does bind to FAT8 regulatory region. Next question was, does the I-1 regulate expression of I-3 and FAT8? So she used short hairpin-mediated knockdown of I-1 in MATAT6 cells. These are the lymphoblast cells that produce IgG. And you can see here, indeed, we have decrease. Green is decrease in expression of I-1, decrease in expression of I-3, and increase in expression of FAT8. So this sort of confirms this part of the network. But the next question is, does this translate to the change in glycosylation and fucosylation? FAT8 is the enzyme that acts fucose. And indeed, Chloe isolated IgG from depleted MATAT6 cells, and we run them with mass spectrometry to assess their fucosylation. And indeed, you can see here that fucosylation significantly increased. Right. So the first part of our talk was, how is IgG glycosylation genetically regulated? And we managed to show that there are 27 regions associated with IgG glycosylation. And we also created this network that suggests how glycosyl transferases and MEN enzymes are regulated through transcription factors and other genes involved in transport of endoplasmic reticulum and GA trafficking, Golgi apparatus trafficking. The next question was, okay, how does this all relate to the various diseases and complex traits from the beginning of the story that we noticed, that they already noticed in 1990s? So to do this, we took all the IgG glycosylation associated SNPs, so the green part of the Manhattan plot. We simply asked, were they already reported to be significantly associated with some other phenotypes in these different databases? And indeed, many of the SNPs that are glycosylation SNPs were already reported to be associated with various other diseases and traits. You can notice some of those that we mentioned at the beginning of the talk. Lupus is here, Crohn's disease is here, rheumatoid arthritis is here, and there are also many more. If you look on a sort of a higher level perspective, there's an enrichment of pleiotropic traits, glycosylation pleiotropic traits in disease ontology category of immune system diseases, which is probably not surprising, given that we are looking at immunoglobulin G, and also the cancer ones are also related with the immune system cancers. Okay, this is all quite nice, but can we somehow incorporate that back to our network? And the question is, yes, the SNPs are basically what we want to know now is the SNPs are shared. So this is the green one is a shared SNP, but there could be two cases when a SNP is shared between two GWASs. It can be a case where their regional associations patterns completely overlap. And this, in this case, suggests that both a disease and a glycan share the same underlying causal variant. Or the other case can be when the SNPs are shared, but sort of they are shared in the tails of regional associations of two different traits. And sort of this reflects the variation, so-called linkage, where there are two distinct causal variants, one regulating a disease and one regulating glycosylation. So we apply this SMR Heidi method that can distinguish between the two on all the diseases we had of summary level statistics access to, and we got the following. So for the glycan trait G0, we did the same for the gene expression. Expression of these two genes, as the expression of these two genes lowers, levels of glycosylation G0 agalactosylated structures is lower. At the same time, the risk for inflammatory bowel disease and rheumatoid arthritis increase. Interestingly enough, this increase of G0 in inflammatory bowel disease and rheumatoid arthritis were already observed in studies comparing cases to controls in various studies. Same, we observed similar with the glycosylation, and there are a few other examples too. But also interestingly enough, for ankylosing spondylitis, height and IBD in these two loci, RUNX3 and IRF1, we show that it seems that there's more evidence supporting that actually there are two distinct causal variants regulating different traits. How does this incorporate, how do we think how this work contributes to understanding why did they observe this changes in glycosylation in these diseases in the previous reports? Well, if you map this back to our loci, we can see here that inflammatory bowel disease are all related with in the region of this 4 genes. So you can now easily see how having a change of expression of these genes could influence in the change of expression levels of FAT8 and could also reflect the change in the levels of fucosylation, or on the other hand, you can also connect it to the B4GALT1. So with this work, what we hope we suggested is we show that there are shared loci between IgG glycans and gene expression and different traits, and then this suggests that the change in expression levels of key transcription factor might explain why glycosylation levels are changed in these specific diseases. This is not referring and not addressing the causality. Are the glycans changed because of the disease, or do glycans are causal for the disease? But I think we have a talk later today, maybe even right after this talk by Olga Zaitseva, that will address exactly this question. So this work was already published in Science Advances. It's freely available, open access. Full summary level statistics are also published in the University of Edinburgh data share. We created 2 additional sort of easier to assess, to look at archives that you can just click through your favorite glycan or gene of interest. And I would like to thank a lot to many people contributed to this project, but specifically Yakov Tsepilov and Yuri Alchenko, that were one of the main drives behind this work, together with people from Genos that quantified glycans and people from Edinburgh that I work with. I'm happy to take any questions.
Thank you, Lucia. We indeed have a couple of questions already. Good. So the first one is from Sodbo Sharapov. He's asking, is there variability of glycosylation of immunoglobulins G produced by a single plasma cell? Or does a single plasma cell secrete all immunoglobulins gene with the same glycosylase?
Yeah, that's a very good question. So they did show that there are people that do antibody-specific glycosylation quantification. So they have plasma cells, they manage to isolate one type of immunoglobulin G coming from one plasma cells, and there is a variability within the one specific plasma cell producing antibody. Yeah.
Another question is from Alexandra Shadrina. How does terminal galactosylation of immunoglobulin G influence inflammation?
So there are some, all of these, so glycans, there's the other side of IgG. There are the effector immune cells that actually respond and react to, sort of the T cell comes and then releases cytokines that actually kill the cell or natural killer cells. And these effector cells on the surfaces have Fc gamma receptor. So these are receptors specific for the Fc section part of immunoglobulin G. And the Fc gamma receptors themselves are glycosylated. So probably it was shown that depending on which glycan is on IgG, the IgG will either stick stronger to the receptor or will bind with less affinity to the receptor, and in that sense it can then introduce a different types of inflammatory response. And also different cells, different effector cells have different receptors that are specific for different types of glycosylated IgG. So there's another way of how to select those cells that will have a specific response. but there are still many studies happening in this area to specifically show how important is galactose specifically, or the whole glycan, or which part of the glycan on IgG are really leading the response. I no longer, I don't know if these are all the questions. I no longer can hear.
Oh, thank you very much, Lucia. Sorry, my mic was off. That will be it. And thank you very much. And we have the next talk coming up in a few seconds.
Thank you very much. Bye.